Schizophrenia, SZ, is a complex brain disorder characterized by cognitive impairments, brain structure and function abnormalities, and genetic factors. Existing diagnostic methods are limited by symptom overlap with other psychiatric conditions, necessitating advanced systems for improved precision. The proposed Multi-modal Imaging Genomics Transformer, MIGTrans, integrates genomics with structural and functional imaging data to capture SZ-related neuroanatomical and connectome abnormalities. MIGTrans employs a novel three-way stepwise attentive integration approach, leveraging complementary information across genomics, connectome, and structural MRI (SMRI) features to enhance SZ diagnostic accuracy. The integration order, genomics right-pointing arrow connectome right-pointing arrow SMRI, is strategically chosen based on inherent correlations between the modalities. The model consists of genomic and connectome encoders, a structural MRI encoder, and a fusion transformer for multimodal integration. The genomic encoder employs a neural network function to learn nonlinear genomic representations, while the connectome encoder uses another neural network function to learn nonlinear connectome representations. A pre-trained 3D dense net 121 is used to extract morphological features from SMRI images, which are then passed to a spatial sequence attention module for learning attention across spatial and channel dimensions. The fusion transformer integrates the learned genomic, connectome, and morphological representations, employing cross-modal multi-head attention to distinguish between individuals with SZ and healthy controls. This fusion process facilitates the integration of diverse data modalities, enabling a comprehensive understanding of the complex interplay between genetics, brain structure, and function in SZ. MIGTrans demonstrated improved SZ classification performance with an accuracy of 86.05%, plus or minus 0.02, offering clear interpretations and identifying significant genomic locations and brain morphological connectivity patterns associated with SZ. The Multi-modal Imaging Genomics Transformer, MIGTrans, integrates multimodal data, including structural magnetic resonance imaging, SMRI, functional magnetic resonance imaging, fMRI, and single nucleotide polymorphism, SNP, data, to predict schizophrenia, SZ, and healthy control, HC, labels. The Conva LSTM module adapts to spatial and channel dependencies within morphological feature maps through equations 5 to 8, involving convolution and elementwise product. The Fusion Transformer module integrates genomic and connectome features through cross-modal multi-head attention, XMHA, emphasizing the fusion of functional connectome with non-imaging genomic biomarkers. The Genomic Connectome SMRI Transfuser module integrates genomic connectome features with SMRI features through XMHA, capturing cross-modal relationships between imaging and non-imaging biomarkers. The fused representations undergo vectorized fusion through dense layers, retaining cross-modality features and improving generalization performance. The feed-forward neural network acts as a classification head, utilizing the fused feature representations to predict target labels. Experiments on the Function Biomedical Informatics Research Network, FBIRN, dataset demonstrate the effectiveness of MIGTrans. The model outperforms single-modality approaches and baselines, achieving higher accuracy through the stepwise attentive integration of imaging with genomic biomarkers. Quantitative and qualitative evaluations highlight the model's ability to identify significant genomic locations, brain connectivity patterns, and spatial regions specific to SZ. Attention maps of SMRI and functional connectome reveal primary regions in the frontal and temporal lobes in SZ samples, aligning with clinical observations. The results emphasize the discriminative potential of MIGTrans, successfully pinpointing SZ-associated SNPs and capturing structural, functional brain abnormalities and genetic factors associated with SZ. The approach surpasses baseline methods, demonstrating its effectiveness in enhancing clinical decision-making and improving diagnostic accuracy. This paper presents a comprehensive review of the current state of multimodal imaging genomics in schizophrenia research, emphasizing the importance of machine learning approaches and the need for further research in this field. The authors provide an extensive list of references, highlighting various researchers' work on topics such as multimodal vision transformers, graph neural networks, and attentional feature fusion. The paper discusses the significance of imaging genetics in schizophrenia research, citing studies that have used functional MRI and connectomics analysis to classify schizophrenia patients. It also highlights the use of machine learning approaches, such as Gaussian error linear units, GELUS, and attentional feature fusion, to analyze imaging genetics data. The concept of multimodal deep learning is also explored, which involves combining multiple types of data, such as imaging and genomics data, to improve disease classification. The authors reference studies that have used multimodal deep learning approaches to diagnose Alzheimer's disease and schizophrenia. An overview of relevant datasets and repositories is provided, including the Function Biomedical Informatics Research Network, FBIRN, data repository and the GWAS catalog. The importance of voxel-wise gray matter asymmetries in statistical parametric mapping, SPM, and the use of graph convolutional networks for schizophrenia classification are also discussed. The authors also highlight the work of researchers who have used attention-based models, such as the Hinge Attention Network and the Gated Contextual Transformer Network, for diabetic retinopathy severity grading and multimodal retinal image clinical description generation. Assuming a high level of technical expertise in the field, the paper provides a detailed overview of the current state of research in multimodal imaging genomics, emphasizing the need for further research in this field.